So thank you, Professor Chin, Professor Wu, for the opportunity to participate in the second ACT workshop. I was fortunate to participate in the first one, and it's great to be in Guangzhou. I love fish, so I hope it's going to be in every meal. So I'm going to spend about 30 minutes to talk about really some of the highlights about phase 1 clinical trials. We've just heard a lot about the ethical issues of phase 1. I'm going to focus more in this talk on the scientific issues, and obviously during the discussion afterwards, we'll be happy to also discuss the ethical issues with you. What I decided to do is to mix a bit of the basics and fundamentals of phase 1 with some points that are more relevant to the contemporary issues in phase 1 clinical trials that we face in 2012. So I think the first thing we should do is really define what phase 1 trial means to each and every one of you. I have 3 slides that I define phase 1 trials, and I'm sure it's not comprehensive. I think the most important element in the definition of a phase 1 trial is that there is a component in your study that has not been answered. It's new, it's investigational, it has not been done, especially with respect to the safety of that novelty. So in this slide, the first examples I've given are phase 1 trials of a single new agent. It could be, for example, a very new agent in the sense that it's even first in kind. It's the first time you have a drug that targets a new molecule in the cancer cell that nobody has done before. And of course, that's exciting, that's novel, but it's also something that means you have to be very cautious because you have no idea whatsoever what happens when you target a molecule that has never been drugged against before. Or it's a first in human, but it has already been tested by other out there, and you're trying to do a similar phase 1 trial, but it's still first in human of your compound. This slide really shows some of the other phase 1 trials, which are more the combination phase 1 trials. So it could be a new drug that's already in phase 1 at the end or in phase 2 with another investigational drug plus an approved drug, or even an approved drug with an approved drug. And depending on the possibility of interaction between these two drugs, obviously you may want to consider doing some PK interaction studies, pharmacodynamics, etc. The third slide are really a little bit different and not your usual thinking of phase 1 trials. The top, for example, is if you really want to understand whether a drug that you're testing has an interaction potential, that when you give it with certain drugs that are already out there, for example, Coumadin or warfarin, there's a significant risk that the patient's bleeding risk is much higher. So you want to understand a little bit more about the pharmacokinetic interaction of that drug. And you might want to give that drug with another test agent that enhances that potential and monitor a patient very closely to see what happens. The second one is really a pharmacodynamic focus. You want to see really whether the drug does do what you think over a range of doses that you're interested in, not just at the highest dose, but at a lower dose, whether the same mechanism of activity is seen. And lastly, for the radiation oncologist in the audience, whether a new drug should be combined with radiation and how to do so in a very specific disease-focused trial is another phase 1 trial. I put in this slide a question that I get asked very often. As a phase 1 trialist, what trials would you like to do if a pharmaceutical company comes to you and asks you what you're interested in? And these, I'm sure, are not comprehensive. I would be very interested if it's a drug that we already have a defined sensitive or resistant population based on very strong preclinical data. And I put in here examples that are very well known to all of you, vemurafenib in BRAF mutated melanoma, or crizotinib in ALK-translocated lung cancer. These do not come very often, unfortunately, to the phase 1 world, but hopefully in times to come we'll see more of that. But the strong preclinical science make them very attractive. Very interested in novel phase 1 drugs that are first in class, something I mentioned earlier. If there's really no drug out there, but it's being developed for the first time with very strong preclinical data again, would be very appealing to a phase 1 trialist. And I've listed in here, obviously, the ERK inhibitor, some of the PD-1 antibodies are very interesting. And I think the third class is something that is a little bit more, needs a bit more thought. for drugs that are already out there, the Me Too drugs, if you're going to develop yet another angiogenesis inhibitor, really why are you doing that is something that you have to consider. Is it because the drug is more efficacious than all the other anti-angiogenic agents already out there, or is it because of less toxicity? And if it is just neither yes to those questions, I'm not sure those drugs should be developed. But if you do have these potential improvements in terms of efficacy or reduced toxicity, so for example, if we're going to develop PI3 kinase isoform-specific inhibitors, I think that would be very appealing because even though we have PI3 kinase inhibitors, a very specific inhibitor would be quite interesting. So going back to the basics, the objectives of a phase 1 study, I put in here the primary objective is really to define the dose-limiting toxicity, and I put in here the recommended phase 2 dose. Many of you are obviously thinking, what about the MTD? I personally have a fear of using the word MTD because to my European colleagues and to my American colleagues, sometimes the MTD means a little bit different. The MTD is in the US and Canada the same as your recommended phase 2 dose, and sometimes in other parts of the world, the MTD is above it. So I like to use the word recommended phase 2 dose because it's clear. That's the dose that you will recommend to your phase 2 trialist colleagues what you're going to take forward in subsequent trials. And of course, we're looking at secondary objectives such as toxicity, pharmacokinetics, what the body is doing to the drug, and also pharmacodynamics, what the drug is doing to the body. And we now obviously look for preliminary evidence of objective anti-tumor activity. I'm going to probably in the future sneak in yet an exploratory objective in phase 1 trial. Perhaps as early as phase 1, we would already want to start looking into predictive biomarkers. And the question is whether we should even enrich in phase 1 trial to look at specific subsets is at this point uncertain. DLT is something very familiar to all of you. Really, it's a toxicity or an adverse event that is considered unacceptable, either because it's too severe or it's moderately severe but irreversible, or it's just chronically irreversible, something that you'll have to live with for a long time, and obviously patients who are sick already do not appreciate that. And we try to accept a certain amount of dose-limiting toxicity without affecting all the patients that we are putting on a clinical trial, because obviously that is not a safe dose. We have very specific criteria that we use to decide what is considered as dose-limiting toxicity, and we also have very standardized language in terms of toxicity grading, so that a grade 2 toxicity of a certain kind, mucositis, for example, in China would be the same thing as a grade 2 mucositis toxicity in Canada. We have the same language, even though we speak different languages. And we obviously have to define it during a certain period of time. Traditionally, we define it during the first cycle of a phase 1 trial, but more and more, I think we're all interested also in dose-limiting toxicity that permeates throughout the entire study. So the late toxicity or the cumulative toxicity probably are as relevant in phase 1 trials than just cycle 1 dose-limiting toxicity. But because you obviously cannot wait for every patient to come off the study before you escalate, traditionally we decide during the first cycle, 3 or 4 weeks time frame, whether DLT has been reached. So examples of DLT, as I mentioned, if you're getting the toxicity chronically or you're giving the drug every day by a pill every day, I suspect most patients would not tolerate a significant toxicity or even a moderate toxicity on a chronic level. So you can imagine having a grade 2 headache every day when you wake up in the morning. To me, is dose-limiting. There's no way we can develop a drug that every morning when you wake up, the patient has a moderate headache, even though, it's not severe. Because the drug will be continued on a daily basis, and they will probably keep getting that headache for the rest of their time on the study. Whereas in the case of intermittent dosing, if you give a drug every 3 weeks or every week, you might be a little bit more tolerant of a higher threshold. So if you have a toxicity that is occurring just very briefly, but it's severe, grade 3 headache, but it's going to last only a day or so, I think some patients may tolerate it, some patients may not, but we may have a little bit more of a threshold to accept in terms of there is a time to recover for such situations. I've listed in here some of the class-specific toxicities as we enter the molecularly targeted era. So not only do we use the fairly generic definition of non-hematological toxicity or hematological toxicity. When we develop drugs of these specific classes, we may even also want to include these into the definitions of DLT. So take an example, for example, in EGFR inhibitors, we know that it will cause skin rash, diarrhea, and nail toxicity. So perhaps you might want to specify in your DLT definition how much of a chronic rash you would tolerate and how much of a chronic diarrhea, despite antidiarrheals, you would tolerate. And some of these are obviously more serious. For example, HDAC inhibitors causing QTc prolongation, which can lead to serious arrhythmias. It's something you probably would not tolerate in terms of a very high threshold. Key concepts, I've already alluded to this. So MTD, MAD, maximum administered dose is obviously the highest administered dose, which may or may not be your MTD. MTD is a bit confusing depending on where in the world you are, and recommended phase 2 dose to me is probably the clearest in terms of definition of your achieving a dose that you're happy with to take to phase 2. And generally that is defined based on the DLT because you want to have a dose that is associated with a certain percentage of toxicity, given the fact that most drugs, in terms of cytotoxics at least, the higher dose you get, you will get some toxicity, and that becomes a surrogate of your activity. This definition may not be relevant in the molecularly targeted era where you may not have toxicities that are dose-limiting, and there is obviously the question where an optimal biological dose when you have no DLT exists. I actually gave a whole lecture recently in Dublin on this, so I don't have time to discuss really in details the OBD, but would be happy to talk to you if you're interested in how an OBD could be defined in a phase 1 trial. So this is a very typical standard 3+3 phase 1 trial where you enter patients at the lowest dose, 3 patients, and you escalate until you see some sort of dose-limiting toxicity. And if you see it in 1 of 3 patients, you expand. And then subsequently, if you see another dose-limiting toxicity, that becomes your maximum administered dose. And the dose behind it or below it is your MTD in the US or is your recommended phase 2 dose anywhere. And that's just really one example of a very simple standard phase 1 design that is still commonly used in many phase 1 trials. The other key concepts in phase 1 study really are the PK and PD, and my next talk will be focusing on that, so I won't during this particular discussion. Now, the key questions: what doses do you start? Typically, there are animal models that guide us, and we use either small models like mouse or rat, or bigger models like monkeys and dogs, and traditionally we would use one of each species to test in terms of guiding us to determine the starting dose of a phase 1 trial. Again, I won't go into details, but there are rules that we follow to do that. And very traditionally, we would use, for example, 1/10 of a dose that would be lethal or toxic to 1/10 of the rodents. Or in the case of larger animals, 1/6 or 1/3 of the lowest dose that would be causing any toxicity in larger animals. And if both of them were done, it would be the more conservative dose that guide your starting dose. And there are tables that help you to convert that dose that you decide into humans in terms of milligrams per meter square or milligrams per kilogram, depending on the way you want to dose. How do you choose your phase 1 patients? These are very standard criteria. We, as you heard previously, we typically test patients with advanced solid cancers that have really no conventional treatments that are of benefit. And we obviously want to have an opportunity to understand the toxicity. So I truly believe in a phase 1 trial, it's very important to pick very good patients with good performance status of ECOG 0 or 1. Putting a patient with ECOG 2 or higher on probably would make your assessment of toxicity extremely difficult. And then there are obviously other very standard criteria in terms of organ functions, previous treatments allow, washout from therapies, etc. Then there are agent-specific eligibility criteria. So if you were testing an HDAC inhibitor that I alluded to earlier, where you know that QTc prolongation is something you're worried in terms of a liability, a risk, you might want to specify what is the threshold of baseline QTc level that you would allow patients to come on, because you do not want them to already be at risk before you even treat them with your study drug. So things you need to think about if you know the class effect or you know something from the preclinical data that makes you worried about this particular drug, and you want to specify that in your eligibility criteria. I want to only briefly touch on the predictive biomarker. Should we select patients in phase 1 trial? And I think Dr. Lee is going to talk a little bit about that in the next talk, so I will not do that. But obviously that is in the mind of us phase 1 trialists, that even though we've discussed that safety is our objective, do we really want to treat all comers? And if we do have a biomarker that already in preclinical models tell us it's compelling, should we limit even from phase 1 to select those patients? And I don't know the answer, and my own decision changes from time to time, but I think a lot of it depends on the preclinical science. If it's very compelling, I would have no problems enriching patients from the first dose level based on that biomarker. And that's actually what a lot of us are doing in terms of molecularly profiling our patients, so that we know exactly what mutations they harbor, and then based on those mutations, we assign them to specific phase 1 trials that are targeting that agent, so that pathway. So for example, if we have patients who have PI3 kinase mutations, and I already know they have that in their tumors, I would probably channel those patients to drugs that target the PI3 kinase pathway for obvious reasons. And that's obviously given that you have the financial support to be able to profile all your patients that you see in the phase 1 clinic. How many patients per cohort? And obviously the 3+3 rule we talked about. There's no magic to 3+3, as I'm sure Jack, our biostatistician, to tell you. It is not too small and it's not too big. It allows you to get some comfort that you know that after 3 patients, for example, if you have 1 patient with DLT, that's 33%. So you want to expand that to 6, so you have a little bit more comfort than if you see 2 out of 6. It's definitely, again, 33%, and you don't want to tolerate that more than a third or equal to a third of patients have DLT, you go down. So it's really a mathematical rule rather than some deep science. And the question of how to escalate as you go from the starting dose to the next to the end, again, is something that has been studied by many, including ourselves, for many years. And the premise really is you want to start off safe. You certainly do not want your first patient on a phase 1 trial to have a severe toxicity or worse, death, because that would not just kill the drug, it will kill the momentum of the team, that you just feel like you've done something really bad. And what you're trying to do is to follow some sort of rule or some sort of model to help you to go up to the dose that you considered acceptable to move to phase 2. And traditionally, we would like to escalate very fast if we know that we are in a very safe zone, and when you started getting closer to your zone that you think it's getting to the dose that will cause dose-limiting toxicity, you want to slow down because you do not want to expose patients to undue harm. That's the premise. And again, there are 2 big groups of phase 1 trial dose escalation methods, the rule-based and the model-based. The rule-based is really, there are rules. You follow the book, and the book says when you finish this dose escalation 100%, the next step you increase by 66%, and then you increase by 50%, and so on and so forth, until you see DLTs to guide you. So the premise that I've mentioned earlier, that you go fast at the beginning because you know you're safe, and then you go slower at the end because you know you're close to the danger zone, is illustrated in the Fibonacci series that I've listed here. And this is the standard 3+3 design. I'm not going to repeat that. People have tried to modify and go outside of the box beyond 3+3 because we really don't want to waste a lot of time if the first 2 or 3 dose levels are really subtherapeutic. So designs such as the accelerated titration design have come up as another rule-based design that may help us to be a bit faster at the initial part, where you only put 1 patient on because you know that dose is probably safe, but when you hit not a DLT, you want to hit only a very medium or moderate toxicity before you revert back to your 3+3 design. So you think at this point you're saving some patients and hopefully maybe some time, although some would argue that it doesn't, but you do escalate with fewer patients to this dose level. And then when you start seeing the moderate toxicity, you go back to your original 3+3 rule and then follow your previous escalation. Those are rule-based designs, and it's easy because we all know the rules, and when you're on a conference call, you know what everybody is going to say because they all know the rules as well. But I think many biostatisticians feel that sometimes a rule is not the right way to do this. And I know Jack is a believer of model-based design because he likes to use all the data, and we don't follow just a rule. And the Bayesian design, I'm not an expert, is really trying to take advantage of the fact that you're gathering all the information from the study and you use it as you go along. You don't just follow a rule. You're trying to actually use what you think is an existent model in terms of the therapeutic toxicity curve. And every time you enroll a patient, that patient adds to help you make that model, make that curve more close to reality. Despite that, there are modifications, for example, to a Bayesian design, such as the CRM, continual reassessment design, because clearly we still have to be guided by safety. So let's say you start off at a dose of 10, and even though the model tells you 400 is fine, it is unlikely that any phase 1 trialist will feel, okay, after the first dose level of 10, let's go to 400. That's a bit too rash, that's too dramatic. So we still set ourselves some rules, but we're trying to use as much information to update the model. And for the model-based design, you do need your biostatistician working very closely with you to help you decide at each dose level after you finish to fit the model to see how much you're closer to the recommended phase 2 dose. This is another version of the Bayesian design called the EWalk, which is another Bayesian design that is actually quite appealing because you're trying to estimate the patients that are going to be underdosed, and that's what you're going to try and avoid. You're also trying to estimate patients that are excessively overdosed or overdose. Again, you're trying to avoid. And what you're trying to do is to get the highest percentage of patients who are getting the ideal dosing. So after each dose level, the toxicity data you've gathered, the DLT versus no DLT in each patient, is fed into this model to help you update to see whether you're closer to here, or maybe you met the perfect model, that is your recommended phase 2 dose. I would encourage you to talk more to Jack if you're interested in Bayesian-based design, because it's obviously very complicated, but it's very, very appealing because you're using a lot of the information you learn as you go along. I put in a very typical phase 1 study we now do in 2012 in terms of a phase 1 study. So no matter what phase 1 dose escalation method you use, you arrive at your recommended phase 2 dose. And very often now, pharmaceutical companies and others are interested in doing expansion. So a tail, the end of your phase 1. Whether you're interested in looking at the pharmacodynamics to show the proof of mechanism that your drug is, for example, an ERK inhibitor and it inhibits ERK, you want to have biopsies or functional imaging to prove that you see target engagement, you see target inhibition, or functional imaging, the PET scan changes, proliferation is decreased, or other signs of cell death. So that's one reason you want to do a pharmacodynamic expansion cohort. Or another reason is you want to start thinking about where to go next. So you're looking at either molecular enrichment. If you weren't going to enrich from the onset, you want to start enriching only at the MTD or recommended phase 2 dose. You might want to do it in the expansion. Or histological enrichment, where you're only interested in certain tumor types. You want to start looking at that. I think it's very important for us to emphasize that the expansion cohort does not equal your phase 2 trial at this point. And we need to understand that these are usually very small groups that allow you to understand a bit more about the dynamics as well as the potential tumor types or molecular profiles that should move forward to phase 2. We did a very short review in terms of looking at the use of expansion cohorts now in phase 1 studies, and we found currently about 36% of patients in trials in 2011 actually use expansion cohort, as opposed to only about 12% 5 years ago. So this is obviously an increasing phenomenon that we're going to see. And we find that actually when you look at expansion cohorts, the best purpose it serves is really to better define safety. It hasn't really helped us do as much in terms of patient enrichment or looking for efficacy. So I think again a caution that if you're going to use expansion cohorts, use it cautiously to know really what are you doing that for, and is not a replacement of your phase 2 trial. I don't have a lot of time on combination phase 1 studies because these are complex studies, because you could obviously dose escalate 2 different drugs in many different ways. And one such design that is very interesting, for example, is this zone design where you can escalate in more than one combination at the same time, where you can increase drug A but keep drug B constant, or increase drug B keeping drug A constant, or increase both when both at increased dose level have been cleared. And as you can see, it's a very complex way to think of how to really combine two drugs safely. And there's a lot more behind this than I can give in 10 minutes or 5 minutes. I think lastly, this is close to my last slide so that I'm keeping myself on time, really is, again, when you do your phase 1 study, think of it, what are the endpoints that you're trying to do. The most important thing is still the toxicity question. Safety and DLTs are your primary endpoint. We do look at other things such as the drugs, levels or metabolism, the dynamics in your normal tissues, what happens, the endpoints, the molecular endpoints, and we look for the preliminary anti-tumor activity. But first and foremost, defining a safe dose to take forward to phase 2 is obviously the most important endpoint. I always end my phase 1 talks with this slide because I think it's very important for us to remember that phase 1 is not just the investigator. And I certainly do not have the ability to run any of my phase 1 studies if I do not have any of my team members, my trial nurses, my fellows are running my practice right now, scientists that help us understand what's going on with these drugs, the coordinators, the labs, biostatistician, pharmacists, radiologists. These are the team, and the only way a trial can be successful in the phase 1 or any setting is really to have a team that works well together and communicates. And if you are running a phase 1 program, at the very least, you should have a weekly phase 1 meeting to talk about all the patients and all the trials in detail so that you all know what's going on with all the patients at all times. Thank you.
Thank you for excellent talk. So, Q&A? Yeah, please, Cindy.
Thank you very much for your very excellent talk. But for the 3+3 the design, it's reasonable for us because we all know that. But for recently, as you mentioned, the 1+1, that is the one patient for the very, very low dose, and then go one by one until the grade 2 toxicity. But if you're convinced that the recommended dose is determined, then you need the PK analysis, so you have to go back to the very low dose of the patient. You have to get the data for a lower dose, middle dose, and the recommended dose. So if you convinced that the higher dose is benefit to the patients, but you put the patients in a very low dose for multiple PK analysis, so is it ethical? I mean, that's the question.
Yeah, I think the accelerated titration design is good when you know that likely that you have a very wide therapeutic index, that at the beginning you probably won't hit a lot of toxicities, that you want to go fairly fast to the second stage where you convert to a 3+3 design. I think in that situation, it does save you some patients that are exposed to very low doses that you're trying to avoid. I think your question is, if you're not sure whether the lowest dose is going to be your best dose, and if that is going to be a question that there is an optimal biological dose with the drug you're testing, then you only have one patient with activity assessment as well as PK and PD assessment. How can you, base all your decisions on that one patient? I guess the question is 3 patients really a lot better? It's a little bit better in that case. And I think if that's your design, that you're trying to get pharmacokinetics and pharmacodynamics to make decisions based on looking back at all the dose levels, and I think using a 3+3 design is probably more sensible, because you don't want to go back and explore the dose levels again later on.
Thank you, Professor Lillian, for your excellent presentation. I have a question about the endpoint about a targeted drug in phase 1 trial. In traditional design about phase 1 trial, we usually need to find the DLT and reach the RP2D or MTD, and we stop the dose escalation. But most phase 1 trial now in cancer field is about targeted drug. So most of the targeted drugs are very well tolerated in patients. So maybe we have reached a very higher dose, and we have seen some response, tumor response, And maybe in cell line experiment, this dose is enough to have inhibit the tumor activity. But we still, the drug is still very tolerated very well. We still didn't ban the MTD. So I want to know, in this situation, can we stop the dose escalation? Thank you.
You're obviously asking the optimal biological question, which, as I said, I had to give a half-hour lecture on before, so it's going to be tough to answer that question in 5 minutes. We actually look back to see all the phase 1 studies of single-agent molecularly targeted agent that have been done in the last 10 years. About 66%, two-thirds of drugs stop because of toxicity, whereas the other one-third stop because of reasons such as PKs, or they just do not define an MTD. Very often the biological agents like monoclonal antibodies, for example, where you just could not see toxicity, and you have to rely on some sort of either PK concentration that in the preclinical setting was relevant for tumor inhibition, or pharmacodynamics like tumor biopsy showing that you have seen, signs of apoptosis or your target being downregulated, and then use that to base your dose. The tough point, obviously, is you don't know if 90% inhibition of ERK will equal to tumor shrinkage or survival benefit in the next 2,000 patients you're going to test. Obviously, if you understand that, it's an easy decision. It's very tough to know in phase 1, based on just preclinical data, how much of an inhibition or readout is sufficient to translate to clinical activity. When I did the review that I did to prepare my talk, it's interesting. When you look back at the approved drugs, the drugs that have already FDA approval, that use this kind of optimal biological activity to define the recommended phase 2 dose, if they just use pharmacodynamic biomarkers alone, like tumor biopsies or some sort of surrogate tissues, it's not enough. So if you see efficacy, I think that's obviously good. If you see tumor shrinkages at every dose level, it's promising. But if you just base it on a pharmacodynamic biomarker in tumor tissue, no drug actually has been successfully do that to define the optimal biological activity. So I would caution, if that's what you're going to use to guide your OBD, unless your preclinical science is extremely compelling, it might be a bit risky.
So follow Dr. Zhou's questions. So in the target so clear, so we need to select with this the mutation patient to phase 1 or the unselected patient.
Should we put patients with mutations on study?
Yeah, in the phase 1 study, because the target was so clear.
Yeah, I think the target, when you do crizotinib, it's clear because you have very good, preclinical science or vemurafenib. But if you look at PI3 kinase inhibitor, for example, we don't really know that the PIK3CA mutated patients, for example, are the only ones that respond. And we're not seeing the kind of dramatic waterfall plots with PI3 kinase inhibitors in phase 1 trials as we do with vemurafenib or crizotinib. So if you did limit it just to PIK3CA patients, you're probably still not going to get your answer. So I think it depends a little bit on the target and the preclinical data.
So, last question.
Thank you. I made a case before, one design of phase 1 study with a new drug plus a standard chemotherapy. But in phase 1, we use a combination. But for the specified, DLT, shall we consider about the chemotherapy toxicity? So, or we just together, no matter what drug?
That's always a difficult question, because how do you know? You're going to expect a certain percentage of toxicity from the chemotherapy, and you have to decide with 3 or 4 patients at that dose level how much your drug has added to the combination. One design I actually have recently used is actually to give the chemotherapy first cycle and see what happens. And if the patients already develop the toxicity, those patients will not contribute to the DLT definition. They will just continue to give the combination, but you only put patients on cycle 2 if they did not develop a DLT based on the chemotherapy alone in cycle 1, if I'm making myself clear. So you wait for the first cycle, and then you only take the patients that do not have DLT and then add the targeted drug on. And I thought that was a pretty interesting design, although you do have to wait 2 cycles before you can determine DLT, and you do screen out a lot of patients who do not tolerate that chemotherapy well.